Bom dia a todos. Muito obrigado para a introdução. Sorry, these are the only words. Most of the rest of the presentation will be in English. Apologies for that. Just let's start right with the sensing strategies as an introduction. You know that traditional field scouting and sampling is laborious and time-consuming, even if you use some sensors or semi-automatic equipment. We know several sampling strategies for precision agriculture. The traditional one is grid sampling, either based on composite sampling or on point sampling on regular grids. If you have some previous information, some ancillary data, you can try to do targeted sampling, so sampling within zones. However, this requires that the ancillary data are correlated with the parameters you are looking for. Both approaches cannot be done with a high temporal frequency. So people came up with the idea to install monitoring plots, just a few one, and sample there with a higher temporal frequency, maybe every year. However, in this situation, you need to have very good selection of the sampling position. And they must be always related to the variable of interest, which is sometimes problematic. So the best approach is to sample with high spatial density and low cost. So in that case you get high spatial resolution and probably you can repeat it also more often in time. We know a lot of sensors that are used in the environmental science, satellites, aircrafts, and sensors in the soil, for example. For precision agriculture, I like to classify this in remote airborne systems, proximal mobile earthbound systems, and proximal in situ stationary sensors. I will mainly talk about these kind of sensors, continuous moving sensors, so a mobile device in front and a sensor with a GPS behind or in front, drop sensor like this. So for selecting sensors according to the sampling strategies, you have to regard the extent, the coverage, and the sampling area or volume that is sensed by a sensor. You need to regard the turnaround time, so that is the time when you have made the observation, when you get the data. Also the temporal resolution you need, data processing, if you want to do it real time or in post-processing. This also defines the use and management, whether you have a predictive approach or where do it in real time, it's a reactive approach. Of course, costs are very important, the robustness of the sensor, accuracy, handling and user interface is also an important point. Another aspect is the combination of data from real-time sensors like this, from the crop sensors, and the use of ancillary data in combination. So this is called online approach with map overlay. This is not often used, but I think this is one of the future applications, and if you make decisions about sensors, you should think whether you can combine them and how combine them. So let's start with source sensors. We can group them in mechanical, chemical, optical, electrical, radioactivity sensors, acoustic and kinematic sensors. There are a lot of sensors which are in research. This is marked with a blue minus. or under development and promising, and only a few are commercially available and accepted. These are with a red plus, like this gamma spectrometer ion selective electrodes. I will start with mechanical sensors, vertical penetrometers. This is an example of a penetrometer, a vertical penetrometer developed at my institute. So 4 penetrometers side by side. There's a first commercial version of an automatic penetrometer by various company, various profiler. It also includes a electrical sensor. However, this instrument is not produced anymore. One problem with the practical application of these vertical penetrometers is the high variability. These are results from the parallel penetrometers from 4 measurements side by side, and you see they're varying very much within 20 centimeters. So people are thinking about horizontal penetrometer, which they can use in a mobile mode without stopping, and there are many presentations and research I have seen on these horizontal penetrometers, but unfortunately there's nothing on the market to buy. So discussion of penetrometers, density soil compaction is extremely important, among others for fuel consumption during tillage. However, measuring is very difficult due to small-scale variability and also due to the influence of soil moisture and soil texture on the soil sensor readings, and there is no sufficient solution for continuous mapping available currently. Most of you have heard about the geoelectrical soil sensors. Electrical conductivity is influenced by many factors. The most important one is clay, and clay affects also other factors like water content, organic matter, nutrient, ions, and so on. Due to this fact, you see multiple correlations with these soil parameters, and it's often hard to distinguish which is the main factor for ECA. There are 2 main principles. One principle is electromagnetic induction, which plays on the induction of a magnetic field due a coil, the coil here, and uses a primary field which is sent into the soil. The soil reacts with a secondary field, magnetic field, which is measured by a receiver coil. You know probably the EM38 and the other sensors manufactured by the company Geomics. They were the first on the market. This is the first sensor. Later they built the EM38DD with 2 coil orientations. This way they get different readings from different depths. And the next stage was the EM38 MK2 with a smaller coil spacing, just 50 centimeter to get readings for even shallower depths of the soil. Another manufacturer is ULM. The good thing about this sensor, it needs no calibration. It's very robust in that respect. It also offers a couple of spacings within this sensor, so you get readings from 3 depths. The other approach is galvanic contact resistivity method. You use 2 injecting electrodes, which put the current into the soil, and you have 2 measuring electrodes, which measure the voltage drop. You can add another measuring electrode, pair of electrodes, and if you vary the spacing, you get readings from deeper depth. The first sensor for agriculture is from Gebbers. This electrodes are arranged along a line, and you have two spacings for the measurements electrodes, so you have two depths of investigation. Later on, the French company Gebbers designed a couple of devices. The RRP03, for example, has a very interesting arrangement. It measures from 3 depths. They have a couple of sensors, so this is an example for RRP06, which is especially designed for sugarcane, having this spatial arrangement of the electrodes. Again, you get readings from the depths. This is a new sensor from Germany, Gebbers company. It measures at 4 depths here, at several frequencies, and it has a gamma ray sensor here on top, so 2 sensors. What you can do with the several frequencies, here 4 frequencies, you can get information about porosity and pore continuity of the soil. Many of you have heard about depth of investigation. It's sometimes a kind of secret how it is defined. So I will try to explain it. These curves show the layer response from infinite height of layer. These are 2 curves. For two has a peak very near the surface, and the wider spacing has a peak at 25 centimeters. If you accumulate the signal, you get curves like this. And the depth of investigation is defined where you have 70% of the signal. So for the wide spacing, the depth of investigation is 55 centimeters. So based on this depth, calculation, you can compare different sensors, and you see that the galvanic coupled resistivity sensors, particularly the Verde, is measuring at very shallow depth, and only the M38NK2 with a small spacing in the horizontal mode is closer to this sensor, for example, from the RRP. So usually you get with these sensors better information about the topsoil. Geoelectrical conductivity sensors are well established. They are fast, mechanically robust, there are no security issues. The EMI is lightweighted compared to this. GCR is cheap. They have a large sampling support to measure a big volume. They can detect soil layering if you have many depths of investigation, and different frequencies might give you additional information. However, relationships to soil properties are ambitious, so you don't know exactly what you're measuring. Some EMI sensors tend to diff, and they are very sensitive to metal and sometimes also to temperature changes. Unfortunately, the resistivity sensors are quite heavy and they do not work on dry soils. So a more recent development is to deploy a gamma ray sensor in soil sensing, and I will explain you. It's based on the analysis of natural gamma ray emission from the decay of radionuclides. The main nuclides are uranium, potassium, and thorium, and it's measured by scintillation counters, so-called. What happens? We have this radioactive active potassium, and during the decay here to argon, it emits gamma ray, and this is measured by the sensor. This is a natural process. It takes place all the time. The decay of thorium is a little bit more complex, and I don't want to get in detail. So a gamma ray spectrum looks like this. We have the main peaks from the three elements, from the potassium, from the uranium, and from the thorium, and you have also background noise and scattering from other elements. You can analyze the spectrum or the peaks, or you can use the integral, all values together summed up. You find good correlation to clay and of course to potassium, sometimes to iron, and it's often very useful to distinguish soils of different geological origin. Most of these instruments are about 100,000 reais. For example, from the Czech company GF Instruments. This is an instrument from Soil Company, which is not existing anymore, unfortunately. Gamma ray measurements are very well acknowledged by scientists. They are fast. You have a direct relationship to potassium content and geology, but only indirect relationship to clay and other components of the soil. Thus, you need careful calibration by reference sampling, and the method is not fully established in precision agriculture, so researchers need how to use it. Ion selective electrodes now are true chemical sensors. You all know from school or from study the pH electrode made of glass. It's basically based on a special membrane which interacts with the protons, and this causes a charge, and that is something you can measure. This is why they're called potentiometric sensors. There's one commercial system available, again from various technologies, and the special thing about this sensor is that it uses a metal electrode made out of antimony. It's essentially the same principle as with the glass electrode. There are two of these electrodes inside this housing, and the soil sample is carried to the electrode by this sampler. On our soils in Germany and on sandy soils we got quite sufficient correlation, but it turns out that you have to correlate the readings from field to field. These are our measurements from the lab, the standard method, and these are the measurements from the on-the-go sensor with the antimony electrode. Here is a map. The blue triangles show the sampling positions of the pH sensor, and the red dots, red and white dots, show the sampling position of a EC sensor which is combined with the pH manager. If you interpolate that map, you get this one. So a nice variation between pH 4 and pH over 7. If you compare that with yield maps, here's one yield map from summer barley, here's one yield map from lupine. You get these scatter plots here. And you should not look for the whole cloud. You just should look for the top of the cloud. Look at the boundary line. And from this line, you can tell the relationship between pH and yield. And you see on the left-hand side, low pH causes low yield, and very high pH, about 7, also causes yield depression. Lupine is not very sensitive to low pH values, so the yield is only restricted by high pH values. Well, we had some problems with the sensor. This is blockage due to residues or loose roots and stones in the soil. But generally, we were very happy because this is a sensor which measures the target parameter directly. There are also other ion selective electrodes for nitrate, potassium. This method is well established in the chemical science. There are no security issues. However, most of these electrodes here are not very robust until you have metal electrodes, but most of these other electrodes use plastics membrane. It's sometimes sensitive to interfering ions. It's a little bit slow. They can exhibit drift, and the more advanced electrodes are much more expensive compared to the pH electrodes. And currently it works best for the protons. There's no electrode for potassium, for example, which would be very, very interesting for agriculture, of course. The next generation is ion selective field effect transistors. This is based on chip technology, so it can be made very, very small. Essentially, it's again an ion selective membrane, which is relevant for the measurements. There are first sensors on the market just for handheld measurements, but you see it's quite a choice you can make. So this one is, for example, for nitrate, potassium, calcium, pH. It's a handheld, like this size. And you put the sample here and measure quickly, and in a minute you get the results. It's made by Hyperspectral Scientific. There's another new sensor development based on IceFed. It's from the consortium Nutristat. It is not commercially available right now, but they want to sell it. The interesting thing, very interesting combination of parameters, so nitrate, phosphate, potassium, and pH. All these in one sensor. However, the disappointing thing, it's too slow, 1 to 5 hours. So the good thing about this IceFed sensor, they can be made very cheap. And there are several options for these membranes, but only a few are available right now. These membranes are sensitive. Also, these sensors tend to drift. And in most cases, you need to construct something for flow injection of soil solution, which makes the handling again complicated, more expensive. Currently, soil scientists are very crazy about visible and diffuse, infrared diffuse reflectance spectroscopy. There are many, many papers about this method. You see here spectra from two contrasting soils. This is a loamy soil, this is a sandy soil. You see the features are not so prominent as in plant spectra. The first design of a mobile VIS-NIR system comes from Japan, Professor Shibusawa. He designed it with a shank, which is in the soil, and you have here a chamber where you put some light in and you analyze the reflection from the soil. Later on, Professor Potzdam from Cranfield University, UK, designed something very similar. So again, this is a shank which is in the soil called subsoiler, and with an optical probe. The recent design of Bonn University in Germany is a little bit different. They have nothing inside the soil. Instead, they have a chamber on the top of the soil, and they measure a very big spot, which is, of course, integrate over a larger area, and you have less small-scale noise. The commercial sensor again comes from various technologies. This is also a kind of instrument with a shank inside the soil, and you have two spectrometers on top of it. Looking closer to the system, here you have the shank. It's in the soil here. Here is a kind of chamber. The light is coming to the soil, it's reflected, and you have some glass fibers bringing the reflected light to the spectrophotometers. You have a shutter for calibration, and you have a sapphire window to protect the interior. Sapphire is a very hard material, like diamond. Soil spectra can be very noisy, so if you have loose soil contact, they look like this, and with firm soil contact they are better off, so you need to do a lot of filtering with this data. Another problem is the sapphire window. Even it's very strong, it can be scratched or it can even break. Thus, because of the complication with this spectrometer system, Veris designed a new system which only uses 2 wavelengths, and it's used only for organic matter sensing. So, the good thing about spectroscopy, it's very fast, you get a huge number of data, you don't need sampling preparation, costs for the vis spectra are moderate, it's relatively robust, no security issues, and there's a long-term experience, and from research, it's currently a hot topic, so it will develop and it will be better and better in the future. However, the problem is there are no distinctive features in the spectra, so there's no direct relationship to the chemical properties. You always need site-specific calibration. Costs for NIR sensors are very high. You need to control the measurement conditions very well, and there's a strong influence of water content on soil spectra. So we have seen a couple of sensors, and I think it's a simple idea to say, let's put them all together. So combine the pH sensor with an electrical conductivity sensor and with a spectral sensor. And we have done that using parts of the Veris system, but this combination is not approved by Veris. So we mounted the spectrophotometer together with the pH sensor and the EC sensor. So you see the water tank for the pH electrodes to be cleaned. Here you see the spectrometer unit, the shank, the fibers, and here you see the rolling electrodes for the EC measurements. Using many sensors, of course, generates a crowdy cockpit. So we have the computer here, especially for the spectrometer, and this instrument is for the EC measurement and the pH sensor. Of course, you need GPS. Can we start the presentation? This is a video, and we'll show how it works. So you need a good tractor with good hydraulics. This is a presentation outside the soil, so this is a cleaning process of the pH electrodes. This is when the sampler is up with soil to the electrodes. This is a downward position for sampling, cleaning at the same time, measuring position. Here you see the hole where the soil is coming in to the sampling stream. And here you see it in action, sampling position, measuring position. You see it's just a few seconds. At the same time, the spectrometer is always in the soil, and also the EC sensor is always in the soil, taking samples every 1 second. This takes between 10 and 20 seconds, the pH measurements. Here you see, it's hard to see, but you see the EC measurements and the pH measurements on the instrument. Okay. So what are the results of combining these sensors? You see here, this is a quality index of prediction. And here you see the different combinations. This is for the spectrometer. This is for the pH sensor. Here, if the EC sensor is included, you have the gray box. If not, it's white. And if it's a gray box, if we have a field-specific calibration, it's a white box if we have calibrated for all the field together. And you see, for example, from the field-specific calibration that in most cases we got the best results if we do it field-specific. And you see that sometimes the combination of all sensors is not producing the best results. In general, we have the best prediction for pH and the worst prediction for total phosphorus. Surprisingly, we got good results for plant available phosphorus and also very good results for total magnesium. But in general, you see the results are only moderate. And the particular point is we always need to do this field-specific calibration. I'll show you some scatter plots. These are the good results. Two fields involved, Red Sea and King's field. So they are looking fairly good. These are the moderate or fair calibration models for phosphorus, for iron. And these are the poor calibration models. So, conclusions from sensor fusion: the simultaneous operation of sensors can cause a lot of troubles. It's difficult to check the quality of the signals, and the sensor can interfere somehow. The next thing is, in the analysis, the question is how to compare and include the different sensors. How should they be weighted against each other? What are the optimum calibration algorithms? And of course the problem is that you need to sample all your fields for calibration. My favorite sensor now is X-ray fluorescence. The principle is based on the excitation of electrons by high energy X-rays emitted by the sensor here. The electrons are ejected from the orbital, and as a response other electrons are falling down to the lower energy orbital, and during that process they emit X-rays, and these are measured in the sensor. The patterns and the energy patterns are very specific for each element, and so this way you get energy spectra with very distinct peaks. You don't have many cases where the peaks are overlapping. This is very good. The point is that you cannot analyze every element. You do not see the so-called light elements with low orbital numbers. So the analysis starts here with magnesium. This is at the edge of the detection. And so-called light filtered elements, they are difficult to detect, and unfortunately they are the interesting elements. And also here, the elements you cannot detect are the interesting ones for us. Potassium, calcium, they are good, easy to detect. You see also other elements, in particular heavy metals. Here it's a slide you should see how an handheld instrument looks like. Unfortunately, it's not cheap. And here you see some results. First of all, I have to point out that you are analyzing total content. It's only the total content of elements. I have arranged them according to the atomic number, so magnesium should be the most difficult, and you see the correlation is not very good. It's not only the atomic number, but it's also the concentration of the element in general that influence the quality of calibration. So you see this soil has a lot of aluminium inside, and you see it gets a good model. Total phosphorus, it's not so good, but probably there are also some outliers due to the reference method from the lab. Here you see for potassium it's not so bad. calcium, very good, and manganese is not so, it is a low concentration level, so it's scattering more, but it's surprisingly good. So the good thing about XRF is you get these distinct features in the spectrum, and this way you get direct relationship to the variable of interest. You got a huge number of data. It's a well-established method in geology. It's fairly robust and you can make it mobile. It's non-destructive. The bad thing, first bad thing is X-rays are harmful. And the operator needs a certificate to operate it in most countries, as far as I know. The other aspect is that it's difficult or impossible to detect light elements like nitrogen, carbon, boron. It's limited to total elements. And it's not so fast. You usually need 1 to 2 minutes to get good readings. And there may be matrix effects. It also has a low depth of penetration into very small spots, so you will have small-scale variability causing noise in data. So you have to repeat it quite often. And the cost for handheld systems are still high, so I think the lowest price is starting here with 90,000 reais. So now, different topic, plant sensors. I group them according to the measurement principle. We have mechanical sensors, probably not so well known here. The most popular sensors are optical sensors. We have heard a lot of talks during this conference on optical sensors. And we have an acoustical sensor, commercially available. So let's start with mechanical sensors and drops. Again, I have to advertise the invention of the ATB. This was our first sensor, later on commercialized by Class company. It's not manufactured anymore, but it's a quite simple principle. It's based on the bending of this pendulum, going back and forward according to the biomass. Another new mechanical sensor is not a mobile one, it's a stationary, it's a leaf sensor. And the interesting thing is that it's related to water potential of the crop. This is a very, very important parameter, very difficult to measure, impossible to measure with optical sensors. So this is a new one, and you can get it from Yarasim company. It's based on the displacement of the sensor here inside according to the leaf thickness. And this is related to the water potential in the plant. So let's go to optical crop sensors. There are a lot of them on the market, which is an indicator for me that they are successful, farmers are interested, and so companies react on this interest and produce new sensors, a whole lot of them. You distinguish these sensors by the number of spectral bands, whether they are passive or active, so come with their own light source, or use the sunlight. You can further distinguish if they have a broad range light source or if they have selective light sources. Also important is the distance to the crop, the field of view and the spot size, the viewing angle. And for the practitioners, it's important how you calibrate them, whether you get support from the seller of the sensor, or if you do or can do your own calibration, and whether these algorithms are free or whether it's a black box system. this is a spectrum of a crop, and this is a full spectrum obtained by a spectrometer. So every 4 nanometers we have a measurement. You don't need the full spectrum, and thus scientists found out that there are some very interesting bands inside the spectrum, and it's sometimes sufficient to use just 2 wavelengths from the spectrum to get good information. In this way, the first index based on just 2 spectral wavelengths is the NDVI, and in the similar way there were a lot of other spectral indices just using 2 wavelengths. The good thing is that you make your instrument simpler if you just use 2 wavelengths, and also sometimes these indices are more robust. So most of the commercial sensors you buy are based on indices, based on 2 or 3 wavelengths. So here you see a more complicated index based on 5 wavelengths. This is the red edge inflection point, and it shows you the position of the transition in this range, where you have a steep increasing slope, and from here on the slope is decreasing again. And this inflection point is very sensitive to chlorophyll content, and it was used in the first commercial crop sensor. Another criterion to distinguish sensors is the bandwidth. You can have narrowband sensors or broad wideband sensors. It has its pros and cons. If you use narrowband sensors, they are more sensitive to noise, but probably the wideband sensors, they are not so sensitive to distinct difference in your crop. The light sources can either be selective or monochromatic, is it? So you have a red or green light, and you have a so-called panchromatic detector, or you have a polychromatic light, so the whole spectrum, and you have selective detectors. Both principles are existing on the market. Another principle is the problem with crop sensors for you as farmers. There's no comprehensive evaluation. It's very difficult to decide which sensor is best, which suits your need. But please understand, the scientists, the comparison is very difficult. And it's not only the sensor itself, but it's also the algorithm behind the sensor that we need to evaluate. This makes comparison very difficult. There is no true N sensor. No sensor measures the N concentration in the crop. What you see is the greenness, and this is related to chlorophyll, and this is often very well correlated with the N concentration. Optical sensors can be distorted, for example, by drops of water on the plant, other weed species between our crop, weeds for example. Drought stress, diseases, and other nutrient deficits can also cause optical changes of the crop and the appearance of the crop. There's always a question about the agronomic calibration, which is the best fertilizer algorithm, how to use these data in a proper way. Optical sensors can be used for other things besides N fertilization, for example for plant protection, growth regulation, in moisture drain for example, or desiccation, that's top killing in potatoes. Another type of sensor not often used in practice are cameras. So we are using in our institute a camera to detect weeds in the drum line. You need an NDVI camera to separate the crop from the plant from the background, and then You can calculate after thresholding, you can calculate the leaf area. And based on the leaf area, you can make decision of the amount of chemicals to be sprayed. This way we achieve pesticide savings of more than 20% without depression of yield. A more sophisticated approach is not only to detect the leaf area, but also analyze the shape and to get information or to decide whether it's a crop or a certain weed. This requires matching with a database, and it's very computationally very demanding. It takes a long time. So this system from the University of Hohenheim needs 1 night for data processing. So it's an offline approach, and it's research only currently. But there is now a commercial sensor, more or less under development. It's called the H sensor. Will be sold by Agricultura, and this is using plant recognition for selective spraying. The principle is essentially the same. You have a camera with 2 wavebands, calculates the NDVI, it segments, and then you make classification based on shape recognition. NDVI cameras are very expensive, and this is why the ATP decided to make a cheap design, a single chip camera, and it's well known in Brazil, as you see already. The features of things is based on a very dedicated red and NIR filter. We use a raw wire pattern. It has a range extended NDVI algorithm, works with high dynamic range, so it's not so sensitive for different illumination conditions. It has a global shutter, so it's good for fast motion imaging, and the cost will be around 1,000 reais. Coming from multi to hyperspectral imaging, I just want to indicate a very interesting new sensor. It's a hyperspectral video camera. It's very small, as you can see, lightweighted, 470 grams. This way you can mount it on a UAV. It takes images, full spectral images, 4 images per second with 128 bands. So this is really a big thing, very interesting. Unfortunately, of course, it's quite expensive. So currently for research only. But they promised me it will become cheaper if you buy a lot of them. So 3D imaging is another way to use cameras. Another example from my institute. It's about precision horticulture. In apple growing, it's important to reduce the number of flowers in apple trees, because too many flowers generate small apples. And we do that tree specifically, and for that reason we need to count the number of flowers. And this is done by a 3D vision system. So it works like this. You have the original black and white image. From that you get something which is called disparity or depth image, which contains the distance information. You can overlay it with the black and white image to extract the white flowers, like it's done here. And from the overlay you know where the blossoms are in the first row. Because we have to look sideways, we always see the next rows on the image. And we need this 3D vision and the distance to just extract the first row. I can tell you that this is a very challenging undertaking and it's not done yet. But this is one approach how to use 3D vision, and algorithms for that are starting to get very fast. So thermal imaging is something which is also of interest for ground stress detection, but it's very difficult to make that under field conditions. Acoustic sensor. This is a new one by Agricultura. It uses multiple echoes to construct a 3D shape of the Colina. Smartphones are now very interesting, and they are full of sensors, as John Stefford already told us. Among them there is a camera, GPS and so on. And there are two applications already available. From Yara, there's a recommendation of fertilization in rain. And another one in corn, also for nitrogen application. It's from Spectrum Technologies. So coming to conclusions. The challenge is that we need to identify the properties of interest directly. And there's still a big problem for our sensors. Most of them only have indirect relationship. The problem is for nutrients and soil crops, don't have a good sense of soil compaction, water potential, not only water content, infections, pests, there are no sensors available for that. So a lot of space for research. For the practitioner, the robustness and the user friendliness is of course very important, the cost and the data interpretation. But it's not only the sensor. Precision agriculture is a complex system and everything is related to everything. So if you don't have the knowledge or if you don't have quality control, the best sensor will be of no good. So you must take care that the whole system is on the same level. Then you can make most of your sensors. So, obrigado pela atenção. Thank you for your attention. Muito obrigado, SPA e Professor José Moura.
Bom, eu primeiro gostaria de agradecer ao Dr. Gebbers pela brilhante apresentação. Eu acho que ele foi muito feliz quando usou o termo hot topic para descrever tanto a área de sensores de solo quanto sensores de plantas, sensores de vegetação. E no Brasil realmente eu acho que é um tema bastante atual, né, e de muita importância dentro das ferramentas DAP atualmente disponíveis e usadas aqui no país. Bom, nós temos aí um momento para discussão, para perguntas. As perguntas devem ser encaminhadas por escrito, né, ao pessoal aí da organização. Já vai chegando então aqui. Bom, uma pergunta do Rafael Siqueira, da Colorado State University. Quais são os prós e contras da câmera em 3D do exemplo da maçã em comparação a sensores de LIDAR?
With the camera, you have already the topology, so the relationship between the observed points. With a LiDAR you have to construct a surface which takes some time. So this is the benefit of a camera. In the camera, the position of the pixels is already known. From the LiDAR, it's very difficult to construct this surface. So cameras can be faster. Disadvantage of cameras: they are more sensitive to the illumination conditions.
Bom, antes, na verdade, de passar a próxima pergunta, eu vi nas minhas anotações aqui, eu havia esquecido de comentar no início da apresentação, o Dr. Gebbers também ele é vice-coordenador do grupo de discussão de sensores proximais de solo dentro da International Soil Science Society. Isso. Apenas complementando aí o currículo do Dr. Gebbers. Passando para a próxima pergunta, professor Molin. Quais as razões você apontaria para não termos penetrômetros horizontais no mercado, considerando que as pesquisas nessa área existem há mais de 20 anos?
I don't have a good explanation. What I have learned is that there are many factors interfering, and so it's very difficult to identify compaction and separate it from other soil factors. And I think there are scientists much cleverer than me have been working on that issue, and so I cannot explain it precisely. There are other approaches, for example, to analyze draft force, which is very simple because tractors have this kind of sensor inside. But also about these sensors, we don't know that somebody is using it. So probably again the interpretation of the data is difficult, too difficult.
Bom, temos mais uma pergunta sem identificação. Como você vê a espectrometria de solos no mapeamento de parâmetros de fertilidade do solo? Ou seja, quanto à presença de nutrientes no solo? Espectroscopia para o mapeamento de nutrientes no solo.
It's currently a hot topic, but we have to be very careful to use this data. So it's on the one hand it's a good option, but on the other hand we must be aware of the difficulties. So we can find good relationship, and then these sensors